Hello, everyone, and a very warm welcome to our webinar today. We really appreciate you joining, and we would today like to walk you through our topic, which is compound absorption by different biochip materials and the consequences on preclinical research. We also would like to thank everyone who already submitted their questions in advance. The reason we did this was that we wanted to shape some of the content of this webinar to what you're actually really interested in, so you can make the most of this webinar. But of course, you can submit questions throughout this webinar. We will answer them at the end of it, and you'll be able to do so either through the chat or the Q&A function, which you can see in your control panel. Also, we are eager to keep this webinar a bit interactive, so we've actually prepared a few surveys for you so that you can chip in as well, even if you don't have a question right now. And in fact, I've actually prepared one right now. So let me bring this up. And hopefully I'll be able to see this question. So right in the middle of the control panel, you see the poll, which is, do you currently work with organ-on-chip models? So easy question to start with. So yeah, please tell us, do you work with them? Yes, no, or are you eager to do so very soon or in the future? Okay, and we've got results in. So actually about 40% of you have worked with organ-on-chip models before, and about 30% don't work with them yet. And another 30% doesn't work with them, but wants to do so very soon. Thank you, that's really interesting. So without further ado, I'd like to introduce our speakers for today. So first up, we'll have Dr. Thomas Sommermann. He's one of our senior scientists, and he completed his PhD at Harvard, where he worked on cell signaling and cell metabolism of lymphomas in Epstein-Barr virus. He continued his research in Germany at the MDC, where he worked on lymphomas, again, in Epstein-Barr virus, with some using transgenic mouse models. And at Dynamic 42, so with us, he's working on developing a pancreatic cancer spheroid on chip model, which you might have seen us talking about on social media already. He'll be followed by our CEO, Martin Rasch, who's co-founder of Dynamic 42. He's been working and researching with on-chip cell cultures applications since 2010. At the Inner Industrial Hospital, and he and his co-founder, they first-hand experienced the need for physiological human organ models that properly represent the situation in vivo, and this is why they founded Dynamic 42. And then since 2021, he's also board member of the European Network for Organ-on-Chip Euroworks. So with this, I'd like to hand over to Tom.
Here. So that should work better. Hello, also from my side. Thank you for the introduction, Juliane. And I will briefly start you up with the model and mostly talk about the biology we're putting in. Martin is then going to talk more about the biology and really the biochips. But as some of you said, they never worked with a biochip. I at least want to give you some idea of what we really want to achieve here and what the whole field of organ chip wants to add to the scientific landscape, to drug screening, to preclinical studies, and what might be a benefit over maybe the classical way drugs are screened nowadays. The first slide that is prepared here is still us, so that I think what Juliane wanted to kind of give a welcome to us here. But anyway, this is us. We are sitting here in Jena, in the middle of Germany. Had wonderful snow weather the last days, and it's unfortunately now again a little warmer. So organ on chip technology. Why organ on chip technology? Why do we need that? I mean, the big idea here is that we have this huge gap between things going into preclinical screening, which we find in tissue culture, and then of the drugs that actually finally make it to the market. You can see this here with kind of all these drugs going into into the discovery phase where you start testing, and then they might have some initial benefits in treating or in attacking your disease. But then step by step through the preclinical research and then through the different phases, phase 1, phase 2, phase 3, and then finally to the patient, you get a lot less drugs getting through till finally the one compound actually makes it to the patient. And of course, the earlier we can detect drugs that could fail or drugs that could really go all the way, the better it would be. Problem is, of course, you cannot go initially right into the human and test. We all know that. So what we do is usually drug screening in 96 well, or of course a bigger well format, looking for the compounds that might work, and then eventually you go to animal experiments, and then you go to the human. But working myself with transgenic mice, a mouse is not a human. We can achieve a lot of things with these in vivo models, and certainly when I did immunology and lymphoma research, there's a lot to be learned there. But of course, if you look really for drug treatment, you really are dependent that the target and that pathway that the target is sitting in is exactly the same between the mouse and the human. And that's too often not the case. And this is why then drugs fail earlier or later than in the clinic then, because the results from the mouse cannot be translated. So we think that organ chips, so modeling a human organ with human cells as good as we can in vitro, making it way more complex in 2D is the solution to that problem. So we try to simulate this complex environment in a very small scale level, produce key functions like a perfusion in a vasculature, so the attachment of the organ into the vascular system, and then really to find these key facts of a tissue. So the 3D complexity, incorporation of all relevant cells, and having a cell-to-cell communication, a direct communication. So in this case here, you're kind of looking at a gut, and you have two options. You either put in different cell types, what is one thing that we're doing. So here, the endothelial cells of the vasculature up top, you can put in monocytes or other immune cells to support this vasculature, and then the epithelial part on the bottom. And here, you don't necessarily even need to put in all various different cell types, but often the cells, if you put in a progenitor type, they will differentiate to different cell types in there because they have the stimulation of the 3D environment of a perfusion, a gut perfusion, which kind of stimulates the gut, the bowel movement going through the gut. And there you get a complexity that you could never achieve in a normal 2D or non-perfused model. This is mostly due to this kind of stretch and strain that you're having. So kind of with the perfusion coming in, the cells get pushed, they get strained, and then you have the shear force of the fluid moving over that. And that's something that a biochip can model and a 2D model could never do. Of course, I also talked about it, the immune competence. That's something you could achieve in 2D, but of course, the idea of letting PBMCs actually go through a vasculature, giving them the challenge that they have to migrate through an endothelium to do their job in a tissue where they are then needed or performing their function, is a much closer thing to in vivo than you would have just adding PBMCs on tissue culture, let them settle by gravity, and then they interact anyway with the cells. Here, they need to migrate through an endothelial cell level before. The endothelial cell lever is kind of also here the one that is one of the really USPs, because it's very different in the different organs. We have different kind of endothelial cells. Every organ has a very specific type of blood vessels. So we can take these blood vessel cells out of the various organs from donors and then put them into our organ chips and then really model the environment of the organ, really there, of the vasculature there. And we can adjust the perfusion rate so that also kind of the smaller blood vessels then have a different pressure as kind of the organs with larger blood vessels going through them. So we can really adjust the conditions here to have it organ-like, and we really see that the cells are behaving very different depending on what you give them as a challenge from the outside of the perfusion. Once you have this model, through having this compartment, you also then can go further and make it more complex. You can add microbes to the gut, which should be sitting there. And with the perfusion, we can kind of make sure that they are not overgrowing, because they continuously, similar to a normal kind of bowel movement, they continuously get flushed out. So we can have commensals in there sitting, and then they really kind of interact with the tissue, and we see the tissue reacting to the microbes there. We can model inflammatory diseases, we can model infection diseases there, and see that how the microbes are interacting with this. Also, with kind of then this 3D setup there, we'll have more molecular gradients. You will have more oxygen coming through the vasculature, then slowly kind of diffusing through the tissue. So we have an oxygen gradient. The microbes will give a gradient of the bioproducts they are secreting. So there will be a gradient in the other direction, something you would also never see in a 2D culture because everything will be in this one layer. So I think the idea of getting more complexity into these tissues and then really allowing for drug screening later on something that's somewhat relevant to what the drug would also see in the human is something that organ chip technology wants to achieve, and we are step by step really getting there to fulfill this promise. We do this in these wonderful biochips, kind of black plastic chip size of an organ of a microscopy slide, and then within these chips you have then these different chambers. Kind of you see this here where you have always a foil kind of separating the top and the bottom. It's translucent, so you can also do microscopy in that. And then in the middle, this is kind of where the magic happens, where these porous membranes are sitting. So they allow us to separate the different cell layers, so the different types of cells, but they are porous, so that depending on the size that we make these pores, either cells can travel or only cytokines can travel, and then thereby we can actually build up the geometry of the tissue. For the biomaterials, of course, this is a very picky story, and it's kind of the topic of today. But this is then something that Martin is gonna really talk mostly about. The idea that what I told you already, this is kind of then how it works. You introduce the cells through these chimneys, and they are connected to the various chambers, and then they are seated in the various channels, and then you can kind of decide where do you want to put your vasculature, where do you want to put your epithelial cells or tissue. And in my case, what I'm doing is then kind of putting tumor cells on one side and then epithelium on the other side. There's really kind of the freedom there to do what you would like to do. Once you have your cells sitting in there and they have attached to the membranes, then you can start the perfusion. So usually, as I said, we would perfuse the vasculature, and then the gut. We would also perfuse then the gut side to mimic a peristaltic motion. But then, for example, the tumor side, we would only perfuse the side of the vasculature, and the tumors would not be perfused as they would be sitting in the tissue. Great thing about our model is that you only need a peristaltic pump. You don't need any other system to buy, so you can get your hand on a peristaltic pump, and then you're good to go. Just to kind of show you what we can do. So on site, we're having a liver model, an intestine model, and a lung model, which we are using for drug screening for companies. So it's the ones we are selling, which are really established in the lab. More or less done, or hopefully soon we will also have done that first tumor model sitting here to be ready for screening. But we've also made costume models, so customers come to us and say we want a specific cell type sitting in there or a specific kind of tissue that we want to mimic, so we can also do that. And then of course we work with a lot of academic partners that all put in their favorite cell types and favorite cell compositions, and then do their specific question there, and then we help them to set it up. But these are then the ones, of course, where we don't necessarily have the protocols available, but these are then within the academic realm, and then you find the publications to that. So with that, I hope I gave you a good idea of what Organ and Chip wants to do. And then after I think a quick survey, as I think is now coming up, we'll have Martin then talk about the biomaterials that we can also really use these for biological screening, which makes it very depending on the environment they are sitting in, and not just of the biology sitting in there. So with that, I hand over to Juliane, and I hope I gave you a good overview.
Thanks, Tom. Yeah. So with this, obviously, we showed you which organ models we like to work on, or at least we gave you a glance on some of them. Now we're actually interested which models you're working on. So we've got another survey poll coming up where we'll be able to tell us, do you work with lung, liver, intestine, kidney, skin, blood-brain barrier, vasculature, tumor, or something else, or multiple of them? So we're actually really curious to see the results of this. So I'll just give you a little bit more time. And now let's have a look. Oh yeah, so there's a quite overwhelming majority of more than 40% working on liver. Yeah, we've actually seen this in the past as well. And then next to that is like intestine and lung, which are about 15-20%. So there's quite a few people working on that as well. And then the rest is sort of like split quite evenly over the other models. So thank you. So yeah, with this, I'd like to hand over to Martin, who'll be talking about compound adsorption on biochips in the next chapter.
Thank you, Juliana, for this nice introduction. And yeah, also from my side, welcome to everyone. After Thomas gave you a nice overview, and I hope that all, also people who are new to the field, are now on the same level. We're going to have a look a bit more deeper into materials and what kinds of choices you might be aware of, and check out before you start to design your own model. So there are a lot of properties for materials to use, and here we listed some of the most important requirements you should check when you start working. First of all, of course, this is a very simple, easy one. Biocompatibility should be a given of the material. Of course, when you look for commercial things available, this is already checked. But if you start with your own materials and your own ideas, be aware of this. Maybe require some samples beforehand and do some own tests if you do not find something in the literature about biocompatibility. Then you should also check if the material leaches into your cell culture. If polymers, especially for soft polymers, can be set free and start to interact with your cells, that can have a negative influence on them. Of course, materials, you can also check if there's a potential for cell ingrowth. Maybe you want this, that your cells interact with the material. It is often desired for hydrogels in complex cell models. Here. And if the material gives you possibilities for chemical modifications. This is a quite nice feature that you should be aware of. So if you can link something to the surface of the material, or if even the material is suitable for micropatterning, so for producing artificial small-scale surfaces that direct maybe your cell growth or cell functions. And one of the big issues we are also addressing today is, of course, adsorption of compounds. Especially this is a very hot topic when you want to use your model, not just to answer maybe biological questions, but really want to use to look into therapeutic options to treat diseases. So compound screening of classical small molecules, for instance, but also new applications like biologics by tri-specific antibodies and so on. What can also be a nice feature is flexibility of the material. And you should also be aware of oxygen permeability, especially when you want maybe to build up models to study host pathogen interaction, and your pathogens need anaerobic or aerobic conditions. You should check this if this is possible with the material. Other properties that have more impact on downstream analysis, for instance, is optical clarity? Can you use your platform under a microscope and check your cells, or do you have to wait until experiment is finished and then be surprised, oh no, there was no tissue anymore. So yeah, this is a good point. Also tunable fluorescence of the material and accessibility of the biological sample. Can you recover your tissue from the device afterwards as a whole or as single cells? Or does an attempt to recover the tissue result in destroying your model? And if you're looking more into scaling up your production because you have a large lab and a lot of people who want to work with it, or you even also want to provide your platform to others, commercial factors come also into place, like how's the costs to produce your devices. It's often also a big issue for academic labs who have to look for funding. How easy is it to fabricate this for your employees? And how easy is the prototyping? So changing geometries, trying different things out. There are some materials that are more suitable for this than others. One material that has been used on a large scale, especially in the academic field, because it is very nice for prototyping, is polydimethyl siloxane, so PDMS in short. I think the majority of you or who are familiar with organ-on-chip applications have heard of this and maybe had already contact with this material. Yeah, it is a low-cost solution for designing own devices, ease of fabrication. It is a very flexible material. Of course, biocompatibility is also given, transparency, and it is also permeable for oxygen or other gases. However, cons can also be the porosity when wanting to work under anaerobic conditions. And if you want to use it in the context of drug screening, you can also run into real troubles because a lot of small molecules especially are adsorbed by this material and hold back and are not available for your biological system anymore. And why is this a big issue for organ-on-chip applications or in general innovative applications that might differ from the common well-plate format? You see here a very nice graph that pictures this. We compared here one of our mostly used biochips to a well of a 48 well plate in terms of surface area. This is largely increased when working with on-chip devices because you do not have just the cell culture area and some sidewalls. You also have like a top, so it is three-dimensionally closed, the culture vessel, and you have some other things. You have channels inside of your chip that you have to account for that increase the surface area, and then a lot of accessories like reservoirs and tubings. All that adds up and gives room space for drug surface interactions, and that can really influence the outcome of your experiments. This has largely been discussed also with the example of PDMS over the last decade already. Here are some example publications for people who are interested in to get more in-depth look. One nice comprehensive small-scale study is the last one from Auner, published in 2019, where they not just looked into 1 or 2 examples, but used a small library of 19 chemicals and look deeper into the issue, what happens with them in different PDMS device geometries. There are, of course, alternatives you can use that are also suitable in terms of cost effectiveness and prototyping. Not all of them here for this count for this, but you have also other options. Commonly used materials that you can use are elastomers, hydrogels, thermoplastic polymers, inorganic materials, silicon, different kinds of resins, and even ceramics is very suitable for cell culture and designing such devices. Here you see, of course, in this table, a large overview of different parameters. Again, the adsorption and absorption highlighted here is in most materials better. So you should have a look into this. And as an example, Thomas mentioned this already, we at Dynamic 42 use solely thermoplastic polymers.
So yeah, and with this, I'm taking over again. Just shortly interrupt the flow and keep you engaged. So you've seen there's a range of alternative materials. So we've got a little or two surveys again to find out which ones you're working with currently. So you will see survey popping up where we ask you which biochip material are you currently working with. PDMS, elastomers, hydrogels, thermoplastic polymers, inorganic materials, silicon, resin, ceramics, other, you're not sure, which is absolutely fine, or you currently don't use biochips. Okay, so we've got some results coming in. So actually about 20% of you are working with, well, no, it's more like 13% now, PDMS. The other 30% thermoplastics. Again, 30% for silicon, really interesting. Some use other materials we haven't mentioned yet. Be actually very interesting to find out what you're using. So if you're free to share, just pop it in the chat. Always interested to learn about other materials. And then, of course, we asked this in the beginning, are you already working with biochips or organochips? So there is quite a lot of people who don't work with it yet. So 44% don't use biochips just yet. Cool. So, and we've got one more survey left. This time you have to just enter a text. So we would actually like to know if you already have had issues with adsorption of your tested compounds in the past, and if so, which compound class and which biochip material you were using in those experiments. We'll give you a bit of time, but you can always actually answer those surveys also in the chat, so you might have seen them in there. So even if you joined us a bit later, feel free to also reply to the polls that we've submitted right at the start of this webinar. Okay, no, just no. So I guess that is someone who has been observing issues or doesn't work with biochips yet. Okay, so yeah, if you've got more answers to this, yeah, feel free to submit them while Martin is continuing his talk. Also, I just want to mention, if any questions have popped up meanwhile while we're speaking, feel free to pop them in the chat. We will answer them at the end of this webinar.
All right. Yeah, quite interesting, but that is maybe a good chance to give you or for you to gather some new input and things to think about before you start working with organ-on-chip, as the majority has not worked with biochips yet. So yeah, Here is a first example using investigating the adsorption of propiconazole. It was also done in this nice overview paper of Aune. And here they saw that after incubating it for 24 hours in a PDMS device, that the ratio to control was only 25%. So 25% of the compound was left in the medium, the rest was adsorbed. So this is quite a large difference to what you maybe wanted to test initially. Of course, when we started having deeper looks into this, we were then also interested how does it look with our materials, and here is for two different kind of chips. An example where we checked for more short-term incubation of 4 hours and a longer one of 24 hours, comparable to what has been done in this paper. And we saw that over 85% of the substance can be found in the medium still after the given time. So this is quite nice. We see a very low interaction here. And so this was a first hint that we can also work with very hydrophobic substances in our platform, or generally speaking, that thermoplastic materials are more suitable compared to maybe soft polymers. And we started then, of course, to look into other other compounds. So here are some more examples, for instance, APAP. And here we looked into 24 and 48 hours of incubation, and the same for torcapone. Both are small molecules. And you see also that we tested here two different kinds of tubings. And what is also obvious that when you use thermoplastics, for instance, you have not really to worry about your chip, but tubing can become an issue because there you can run still into some trouble. So you should also check this material. We made some very nice experiences with using silicone tubing that showed so far less interaction for the compounds we tested. Yeah. So usually, generally speaking, we see less than 30% of a compound to device material interaction. And of course, also other molecules are getting of interest for looking into organ-on-chip applications because, of course, they are interesting for new therapies. So a big topic over the last years has become prominent with therapeutic antibodies, monoclonal antibodies, as well as bi and tri-specifics. And here you see an example of 3 antibodies with their corresponding isotype controls. And unless one isotype control, a lot of the antibodies tested show very nice results. So also for biologics, you have low adsorption rates with thermoplastic materials, for instance. And yeah, so here also it is in our case less than 30% after 24 hours of perfusion in the whole setup. So to sum this up, this short introduction into things that you should be aware of when choosing materials is that, of course, soft polymers are quite nice to use when you want to do rapid prototyping, and on a low-cost scale, building up your own biological models, investigating biological questions. However, when you want to go further and see implications of treatments to new therapies like small molecules, antibodies, or nanoparticles, then you should consider other materials as options, because with soft polymers such as PDMS, you run really into trouble. And thermoplastics is one option you can use. We at Dynamic 42 made very nice experiences with such kind of materials. And what we also saw, tubing material can become an issue. We tested different ones. Among silicone was also PTFE and other things. And with silicone, we made the best experiences in this case, in this area. And with this short introduction and overview into the topic, I want to thank you for your attention. For those who do not know us yet, here's a small glimpse how our team looks like. And with this, I think we're open for your questions and go into the Q&A session.
Yes, absolutely. Yeah, thank you, Martin. Thanks, Tom, for your presentation. And yes, now we're open for questions. So obviously we've got the ones that you submitted in advance, but feel free to pop more into the chat and we will try to get back to them as much as possible. Also, we will be sending you the recording of this talk. So if you've missed part of it or would like to reread some of the slides, don't worry, we'll be sending to you afterwards. Okay, so then I can't see any questions right now in the chat. So how about we move to our first one that we got submitted in advance, which was, I'm specifically interested in your liver models and would really appreciate if you can talk about this model in 1 or 2 slides. Okay, so that's an easy one to start with, to give you a bit of an overview, and seeing that I think about 40% of you are interested in liver, I think that's a really good one to talk about. So I hand over to Tom and Martin for this.
Yeah, I'll briefly talk about the liver model. It's one of the models we have set up here, so we are running in-house, and it's what you kind of can see here in this closure graph, a system set up of a two-chamber system, so with vasculature up top, and then the hepatic compartment on the other side. We put endothelial cells in the top channel and perfuse it then with medium for the vasculature, and then we below have a non-perfused channel where the hepatocytes are sitting. We can make this more or less primary, depending on what your setup is required of. We can have, usually we would use endothelial cells from the liver, so liver sinusoidal endothelial cells, and then underneath we can have either primary hepatocytes, but usually we use a saline, which still has a huge potential of doing all the metabolic activity that a liver should be doing. You see here kind of then the idea of immunofluorescence in the end of the vasculature. We have monocytes sitting in there, so some of the monocytes are also then can be found then sitting or integrating into the vasculature, and then we have the hepatic compartment on the other side, which is expressing a lot of the standard hepatocyte markers. So they are making albumin, they are making urea. We have a much higher expression of the transporter proteins and metabolic enzymes that are found in the liver. So MRP2 or the various subs we are looking at are much higher expressed in this then 3D structure, where especially the interaction with the endothelium on the other side gives a huge boost to expression of the hepatic enzymes and the hepatic transporters compared to a 2D culture. Also, when we look at this in a microscopically or in the electron microscopy, they look much nicer once they're having the perfusion on the other side and are having the interaction with the endothelium, and also to some extent with the monocytes, which make it even nicer as they're kind of for tissue integrity. As immunologists, we're always very happy to see how the immune cells are actually also not just providing immunity to infection, but also helping for tissue integrity, which we can also see in these models as they are interacting with the tissues. We do see then also when we can do studies with this, something that I just picked out now of some of the presentation slides we're having, and if you have specific more interests, we can also certainly ask more specific questions. But for example, we can treat them with LPS. This is kind of then one of the inflammatory models we can do, and we see that monocytes, which are here now stained in green, are interacting more with the endothelium as they're getting stimulated with LPS, and we also see them moving into the hepatic compartment in a more active state. And then there we can also see the inflammation coming up. So if we then model cytokine secretion on the hepatic side, where we can sample during the experiment, we can sample the supernatant in the lower channel of the hepatocytes, and then we are seeing the interleukin and TNF secretion here. This should actually be videos, I'm told, but I think I can't start the video. So it's very pretty, actually. If you see this, you can do live microscopy, and you can actually see the monocytes moving. But this is kind of here with the webinar presentation mode. For some reason, I can't interact with this. But maybe also a teaser to another question we're seeing there. Of course, you can do live microscopy, so you can see immune cells moving. If you stain them beforehand, you can see them integrating into tissue. It's very pretty to look at, and a wonderful technique where you kind of, without having a tremendous kind of in vivo imaging or mice or so, where you can actually see immune cells interacting with the tissue. So with this is kind of the idea of what we can do with the liver. We can do infection studies, we can do inflammation studies, and then certainly the tox screening. Usually then we would look for stress markers on the side of the hepatocytes as they're reacting to toxins or so. So maybe with that, I don't know where the question was coming from. I hope this helps already, and I would then move on to the next question, or how are we going to do this, Juliane?
Yeah, we've actually got a few in the chat now, which I would do before we go to the next one that was submitted in advance. So actually, Henriette is asking, how about your biochip? How are your biochips produced? Did you send your specific design to a company that is producing the chips for you? So I think that's a nice one to send over to Martin to answer.
Yeah, thank you for this question. And yes, our core field is doing organ on chip and providing meaningful models for applications. So this one is outsourced. We are focusing really on applications in biology. We do in-house the design and prototyping, and once this is set up, we have a partner who kindly supports us here doing the large-scale production. This is simply a strategic decision, of course.
Thanks, Martin. And there's another one, which is, what was the experimental setup when biochips were tested for adsorption, and was it tested in static or dynamic conditions?
All these tests we used so far is that we, of course, looked in dynamic conditions, the perfused setups. When you want to look into specific parts of your setups, like the chip itself, the tubing itself, the reservoirs, of course, you have only just the option to do this in a static way. But when you look at the whole system, then we always do the cell-free perfusion setup. And here you can even distinguish a bit more, or just dissect your approach a bit more. You can do a complete free setup of biological components, or you can also have a look into what is still adsorbed by coatings you might apply to your setup. So speaking really of a cell-free setup, but doing everything else as you would do it in your biological model. So also applying coatings before you do the adsorption test. This can also be quite helpful to have a deeper look and understanding your setup.
Perfect. Thanks, Martin. So I think we'll be moving to our next pre-submitted questions. We've got more on the chat. We will get to them after that. So Tom, do you want to continue with the next one?
So we had a very specific question for the lung-on-chip model with the exposure system where you can put in aerosols. I briefly had a look at what it's doing. I think it's done by a Fraunhofer Institute, which is for the people not familiar with it. And so maybe for that one person in the chat that exactly knows how this works, I had a brief look. I think there is no reason why this shouldn't work with our system. So I guess with this exposure system, you're getting an aerosolized infectious reagent, and then you can put this into a certain chamber system where then it interacts with the cells and you get an infection really through the aerosol and not through a liquid phase. I don't really see a reason why this shouldn't work. Of course, our system is all set up on a Lure plug system. So you could, if you have an adapter, plug in the Lure plug into your ventilation or in this exposure cube system, and then it will go through the air interface of our liquid air interface lung system, and it will interact there with the cells. Of course, in the end, it will all come out on the other side, so you have to put in some filter system, either kind of going back into your Expo Cube system or somewhere trap the agent, as you probably don't want to escape this into into the lab space. But generally, there's no reason why this shouldn't work. You're accessible; it's all Lure plug compatible, so you will be able to add whatever you can do there. Of course, at this point, it's only Lure plug, so it's not Lure lock or anything. So you will have to be sure that it's really kind of sealed within itself. So I don't know how kind of this then interferes with BSL-3 or BSL-4. So I'm not so sure how you could do this there. Of course, it's not completely sealed, and also, of course, you will have to have a look at kind of the dynamics in there. We are, of course, optimizing this for flow dynamics. So we all have the kind of the data how the flow will be in there, and the cells get very evenly distributed in there, so we don't have too much perturbation inside of the chamber. How this would look like then under if kind of the flow of your particles there in the aerosol would have to be tested. I wouldn't be 100% sure whether you get the same infection rates in the kind of early chamber compared to the back part of the chamber. So that's something that you would have to test out. But I would be excited to have a look at this, of course, especially our lung department. They for sure would be interested to look at this. So I think technically, yes. How then you have to do this, you would have to figure out yourself.
Thanks, Tom. So I will take another one from the chat, which is, I'm wondering if the system is compatible for coating layers with ECM proteins, and how the cells can be seeded in the system, and how time-consuming is the process, and if we provide pre-seeded chips with cells. So
I think
handing that over.
I think I'm gonna take care of that. Really, talking maybe more from the lab side about this. So first thing, coating layers. I mean, we regular coat with normal kind of standard coating techniques for various cell types. So then you get some basal lamina where the cells are sitting better than just directly on the membrane or so. So that's no issue at all. But you can make this also as complex as you want to. So I am regularly using 3D gels to really fill out full chambers to have a complete 3D gel in there for cells to culture, especially in the tumor environment, right? You get the tumor microenvironment that you can only model if you really put this into a 3D matrix-like structure, and this is easily applicable. So of course, depending a little on the matrix you're using, you have to be careful that you're not putting in bubbles into the system, so it needs a little tweaking, and it's certainly not something you will do completely okay right with the first try. But with all the gels I tried, after a few tries and you handle how to kind of get rid of the bubbles, it's very easy to apply. And that's mostly because it's so easy to accessible. So for normal cell seeding, as you're asking, it's super easy to get the cells in. For somebody who has done tissue culture before, you use the normal tissue culture techniques, pipettes, normal Eppendorf tubes or so, where you prepare your cells, you go in with your pipette, you pipette them into the chip. The fluidics are all laid out that the cells will be evenly distributed over all the membrane, and then the cells will just settle and seed there. So after trying a little, and we do this with new customers, we usually do this first here on site, where we invite people for an academy where they can train this. So if you've done it once or two or twice with somebody who knows what he's doing, you can very quickly adapt the system. So from that side, or from that perspective, it's very easy to get into. It's not high throughput in the end, so we're not talking that you can run 100 chips or so. So it's really kind of the idea of running a handful of chips. So maybe up to 20 cavities or so is something reasonable. One can do once it's really experienced, but more is not applicable. So this is really then something that the system is not made for. I hope that answers the question. So
perfect. Thanks, Tom.
Yeah.
Yes, so I will take another one that was pre-submitted, which is how can we effectively address organ growth dynamics, such as like turnover of cells upon progenitor expansion or differentiation after injury or drug treatment, involving multiple organs using organ-on-chip technology.
So, of course, to address this question, we would like to use this chance to show you also general possibilities you have, but these analysis techniques help you also to answer these questions. So basically, you can do a lot in investigating your system, what you already know from a normal tissue culture, like supernatant sampling, where you can then look for different kinds of molecules, like typical clinical parameters, cytokines, compound turnover. But these parameters or biomarkers can also help you in estimating cell growth and tissue dynamics inside. Then, of course, you can also recover the tissue. This is another option, but you can also, when we are looking at the specific question, maybe something like live cell imaging is more suitable to address this question, where you can also look for different parameters, and maybe in this case, fluorescence labels might become in handy to track your tissue dynamics. Also, or you look into enzymatic activities. And here, maybe for this kind of question, it would be quite nice to use an assay that shows you something about cell viability and cell numbers. Something that we recently tried out here and made first good experiences is, for instance, the VST8 assay. It's an assay about viability, and that you can also adapt with some standard curves to cell numbers to get an estimation of cell numbers. And what is quite nice is that it is fluorescently based, and the compound is formed in the cells but not trapped in the cells. So it is released. You can just sample supernatant to analyze this and don't have to destroy or sacrifice your model as you would do as an endpoint analysis. So you can use this assay, for instance, to measure in a timely manner at different stages of your model.
Thanks, Martin. So take one again from the chat, from Daniela. So she was just wondering why our chips are black, actually, and if we could produce any transparent ones. I actually asked that very same question when I started here a couple of months ago. So I guess I hand that over to.
Yeah, so we are working also on using more transparent materials. This can of course be done. The reason why we use black, of course, it's cool. It's nobody else uses it. Gives the chip this kind of Batman optic. But the main reason is we have special production requirements that make it just easier going for the black material. And I mean, the cavities are transparent with the bonding foils and the membrane, so you can easily check the model using cell culture microscopes. This is possible, but yeah, we also want to do transparent chips, and our engineer is currently also digging into this topic and made some nice progress. So stay tuned. In the future, there might be something coming up.
Very interesting. Thanks, Martin. So we take a pre-submitted question again. So could you please highlight ways to integrate mechanical stimulus as a factor in organ on chip studies?
Yeah. Currently you have a few major possibilities. Of course, once you're already applying mechanical stimulus by the perfusion of the models themselves, because as long as your chip design is not completely rigid, if it has some flexible parts, also the perfusion already introduces some kind of deformation. Depends on what kind of flow rates and also perfusion device you're using, if it is more controllable or less controllable. With peristaltic pumps, of course, you can already make use of the peristalsis and maybe adjust this a bit to more physiological levels. And then you have already this input in your model. What can next to this also be done is an more active stimulus to deform the whole device. For instance, by providing overpressure or vacuum, works in both ways. But of course, for this, your whole device or the main parts where the biology happens have to be flexible, and you have to be aware of it is invasive technique. So you're applying some additional component directly into your biological model. And there are, of course, some other ways to do this a bit less invasive, where we also have an internal project running to develop something like this. But here I cannot tell you any more about our own development because it will be something completely different compared to pressure or vacuum.
Thanks, Martin. So in the interest of keeping in time, I think we should just do one more questions before we conclude this webinar. And I would actually like to take one from the chat from Shadi, asking, I'm interested in multi-organ. Do you possess a multi-organ on a chip system as well? What are the most common organs for integration to enhance metabolism and drug screening studies?
So first about the multi-organ system. So as I said, these chips are all the plug system put together. So you could, in principle, add as many organs together as you want. We usually do not more than two. I mean, we are aware. So usually the idea would be, for example, a drug application through the gut, and then you see the metabolization in the liver, or a drug application going through the blood vasculature into the liver, and then you see the metabolites going to some other organ to see whether you get tox or something in this other organ. So this works nice, but we know that competitors are going for the idea of like the whole human on a chip, and we intentionally don't necessarily want to do that. I mean, of course, if you're a academic partner, we are happy to support you in this to go as complex as you want. But what we are seeing in the lab is that already having the single cavity built up so that it's stable and reproducible, kind of giving you results that you can really kind of bank on and show and publish. It's already complicated enough to do that in the single organ. And it's not something where it's 2D culture and always works the same. So it is a complex organ, and there's for a reason that we do this on site for drug screenings, because it needs expertise to kind of build this up consistent and always the same. And if you add additional chambers, it gets more complicated. And it clearly it's possible to set up 2 organs and maybe 3, and we do this here. But the more you're adding to this, the more complex it gets. So really, if you think about the whole human on a body, you need all the cells the same time as you're seeding them and the right passage number. Kind of everybody needs to be happy and healthy. And then the interaction, perturbation through the interaction of the cells becomes much bigger the more organs you're adding. So we want to kind of provide the real idea that we can do one organ by itself as stable and really consistent results, so that it's really doing what it's supposed to do, and then you can add a second one. But once you're adding more and more, I think this is not really where the technology is at the moment. So yes, you can do it, but I would rather do 2 and not too many organs at the same time. And then I am not exactly sure what the metabolism part is, but clearly, I mean, the idea of always looking at drug metabolization in the liver and then seeing how it performs in other organs is something that we regularly do. And of course, that's the benefit of being able to hook these things together, so that you could put a metabolic part of the liver or kind of this uptake part in the gut together with other with other organs, so that we really follow the drug, how it's coming into the body, and not just throw it onto the cells and see what they're doing. I can only say from my cancer side, I mean, it's dramatically different whether you directly put the cancer drugs directly on the cells in a 2D culture. They behave very differently once we're perfusing this through the vasculature, where it usually would be coming from, and then it looks very different. So having more cells between the target and where the drug is coming from is certainly necessary.
Perfect. Thanks, Tom. That actually sounds like a nice topic for another webinar. So with this, I'd like to conclude this webinar. Thank you everyone for participating today and being so interactive. We actually have too many questions to go through them now, but we will definitely answer them afterwards. We will get back to you, and should you have more questions, feel free to also submit them after. We've also sent you a recording, as mentioned before, so you can revisit our slides. And then, yeah, thank you for joining today, and we hope to see you again in one of our future webinars. Goodbye.
Goodbye. Thank you for attending. Hope to see you soon.